All right, this is standard 1E. Standard 1E covers the mitotic cell cycle. So to start off, every cell cycle starts off with interphase, okay? Interphase is going to be the longest part of the cell cycle. Interphase is used for growth and DNA replication. And that's why it's the longest part of the cell cycle. It also includes two checkpoints, the G1 and the G2 checkpoint. The G1 checkpoint is going to check for DNA damage. The G2 checkpoint is going to check to make sure the DNA replicated properly. G1 checkpoint starts interphase. G2 checkpoint is at the end of interphase. Interphase has three subphases, which are G1, S, and G2. G1 is the phase where the cell grows to prepare for DNA synthesis. The S phase is where your DNA is actually going to replicate. So during the S phase is where you have one chromosome initially made up of one piece of DNA, but during the S phase, that one chromosome is going to be made up of two pieces of DNA. So you go from having unduplicated chromosomes to having duplicated chromosomes during the S phase of the cell cycle. Following the S phase, you're going to have G2 phase, which is when the cell is going to grow to prepare for division, and also this is when your centriole is going to replicate. Remember, centrioles are found exclusively in animal cells, and they are used to make the mitotic spindle for cell division. Now, following interphase, if the cell passes all the checkpoints, it'll advance into M phase, which is a period of division. But if it does detect damage, the cell cycle should stop and try to repair itself. If it does not repair, the cell should undergo apoptosis or programmed cell death, okay? If the checkpoints are violated, damaged, or if the cell skips them, then a damaged cell can progress through the cell cycle, and this usually leads to something like a cancerous cell being produced. So we don't want that to happen. The M phase is the second phase of the cell cycle, and the M phase is a period of division where first the nucleus divides and then the cytoplasm. In mitotic cell cycle, the M stands for mitosis, and mitosis means to divide the nuclear contents once. Mitosis is broken up into four subphases and has one checkpoint. The four subphases are prophase, metaphase, anaphase, and telophase, and there is a M checkpoint which comes to the end of metaphase. Prophase is when the nucleus breaks down, the nuclear membrane breaks down, so the nucleus disappears. It will not return until telophase. Prophase and telophase are the opposites of each other. So during prophase, the nucleus breaks down, the spindle forms, and DNA actually condenses. When your DNA replicates here, it's called chromatin. It's relaxed, and you cannot tell one chromosome from the other one. It looks like a bowl of noodles. But when you get to prophase, it actually shortens and thickens where you can tell the difference between the chromosomes. And that's when you see that familiar X shape. Following prophase, you have metaphase. Metaphase is when the chromosomes line up in the middle of the cell. At the end of metaphase, you're going to have that M checkpoint, and that M checkpoint is going to check to make sure that each chromatid, because each one of these pieces of DNA is called a chromatid, it's going to check to make sure that each chromatid is attached to a different side, a spindle coming from a different side of the cell. Your centrioles migrate to opposite sides to form that spindle. So you're going to make sure at the M checkpoint that each one is attached to a different side of the cell. During anaphase, the things that are holding the spindle fiber, holding the sister chromatids together will be cleaved, and the sister chromatids will migrate to opposite sides of the cell. So that in telophase, a new nuclei will form around each set of DNA. The spindle breaks down, and the DNA is going to relax back out. This needs to happen so that when the cytoplasm divides doing cytokinesis, each new cell will have an identical copy of the DNA. DNA replicates to make sure this happens, and then during anaphase, it separates those sister chromatids to make sure that each cell gets a whole copy of DNA, all the chromosomes. Now, cytokinesis is when the cytoplasm actually divides. You don't have two cells until cytokinesis. In animal cells, it's going to form a cleavage furrow, so it goes from the outside in. And in plant cells, you're going to get what's called a cell plate, so it goes from the inside out, because you can't just pinch off a cell wall. You have to build a new wall between the two nuclei. So, if we look over here, the purpose of mitosis is growth, development, repair, and asexual reproduction. So mitosis is going to produce clones. All the cells are going to be exactly alike. Now, with asexual reproduction, we see this with vegetative propagation, budding, and with, that should not say binary fission, that should say regeneration. Regeneration is the other one that is done through mitosis. Now, bacteria also reproduce asexually. and bacteria use binary fission. And the reason why bacteria use binary fission and not mitosis is because mitosis means to divide the nucleus, and bacteria are prokaryotic. They don't have a nucleus. You can't divide what you don't have, okay? So bacteria use a process called binary fission, which is very similar to mitosis, but it is not mitosis. Now, during mitosis, my parent cell here is diploid with 2n equals 4, so there's two different types of chromosomes. I have 4 chromosomes made of 4 pieces of DNA. When the cell enters the cell cycle, it's going to go through interphase. During interphase is where DNA replicates. So again, 2n still equals 4. There's still only 4 different types of chromosomes, but these 4 chromosomes are now each made up of 4 pieces of DNA. So here I had 4 chromosomes made of 4 pieces of DNA. Here I have 4 chromosomes, but I have 8 pieces of DNA. So the amount of DNA doubles, not the number of chromosomes. Following interphase, the cell is going to enter into prophase. During prophase, the nuclear membrane breaks down, the centrioles migrate to opposite ends, and the spindle begins to form and will attach to each side of the chromosome, making sure that each chromatid is attached to a spindle fiber. During metaphase, they're going to be lined up in the middle of the cell. Anaphase, the spindle fibers begin to shorten, which separates the sister chromatids from each other. During telophase, the nuclei reforms, DNA relaxes, and the spindle disappears. And then during cytokinesis, you split into 2 identical cells. One way you can pronounce mitosis is mitosis, which lets you know you get 2 sister cells at the end of mitosis. Toodles.